Pain management and the treatment of inflammation are important considerations following operative procedures. Appropriately managed pain allows for patient comfort and avoidance of increased costs associated with undermanaged and persistent pain. The Regenesis Provant therapy system is an option that is non-narcotic. Patients can self-treat at home with simple twice-daily treatments. Provant works by delivering a field of electromagnetic energy that changes ion gradients across the cell membrane, leading to the activation of secondary messengers. The result is a cascade of gene expression and protein activity that brings inflamed tissue through and out of inflammation more rapidly. These secondary messengers have varied effects, including increasing anti-nociceptive analgesia by inducing endogenous opioids that turn off pain signals at nerve ending, reducing edema by upregulating the expression of aquaporins, and up or down regulating a wide variety of other inflammation-related secondary messengers, resulting in a more rapid resolution of inflammation. Regenesis Biomedical's Life Science Laboratory researches the effect of Provant on human cells in an effort to better understand these mechanisms of action. Our findings show that the Provant electromagnetic field changes ion gradients on the cell membrane. This influences the movement of potassium, sodium, and calcium ions across the cell membrane. These movements change the membrane potential, which activates secondary messenger systems and effectively acts as the receptor for Provant therapy. Examples of second messengers affected include nitric oxide synthase and calmodulin, and potentially several others. These second messengers translate environmental stimuli from the cell membrane to the nucleus, stimulating the creation of transcription factors. These transcription factors regulate a number of genes that either inhibit or increase the production of certain important proteins. One such group of proteins is interleukins. Interleukins are cytokines that mediate communication between cells and stimulate immune responses and regulate many of the processes during inflammation. RNA strands that form pro-inflammatory interleukin-1 beta and tumor necrosis factor alpha are downregulated, while RNA strands that form anti-inflammatory interleukin-10, lipooxygenase, heme oxygenase, and cyclooxygenase are upregulated. The RNA strands create their associated protein molecules, and the net effect is that Provant upregulates interleukin-10, an anti-inflammatory cytokine, as well as anti-inflammatory lipooxygenase, heme oxygenase, and cyclooxygenase. Growth factor VEGF is upregulated. These are just a few examples of proteins that are regulated through the use of Provant therapy. Provant therapy also regulates genes that enhance the anti-nociceptive pain feedback loop in inflamed tissue, reducing patient pain and discomfort. Provant relieves local pain by affecting gene expression that regulates the endogenous opioid pathway. Provant stimulates the increased expression of ET1, while also increasing the expression of ETRB receptors on the surface of the keratinocyte. The result is more ET1 binding on the keratinocyte and an increased release of endogenous opioids. This includes beta-endorphins, enkephalins, and dynorphins. As more opioids are released, more are received at the pain fiber. This causes more potassium-releasing Gurk channels to remain open, and the nociceptor's ability to transmit a pain signal is reduced even further or stopped. The result is pain relief for the patient. While pain relief is critical, the successful treatment of inflammation involves more than just reducing pain. Inflamed tissue can become edematous, and this buildup of fluid can hinder the resolution of inflammation and pain. Aquaporins are proteins that form pores in the cellular membrane. They selectively conduct water molecules into and out of the cell. During tissue inflammation, aquaporins help regulate edema. Gene expression regulated by Provant increases the number of aquaporins in many of the cells involved in resolving tissue inflammation. The increased expression of aquaporins facilitates the exchange of fluids between and amongst various cells in and around the inflamed tissue, expediting the resolution of edema. The resolution of inflammation involves a complex cascade of overlapping activities that involve and are governed by gene expression. During resolution, specific genes and proteins are required to rebuild the tissue structure, which supports new cell growth. Growth factors and extracellular matrix begin this rebuilding. This allows the cells to go through their cycle of growth and division, providing tensile strength to resolve the injury. Provant increases the key growth factors VEGF and PDGF and increases collagen, a primary component of the extracellular matrix. Regenesis Biomedical's Life Science Laboratory continues to research the various ways that Provant influences gene expression so that we can better understand the mechanisms of action that it influences. In summary, the management of pain and inflammation is an important consideration following any operative procedure, both for patient comfort and to avoid increased costs associated with undermanaged and persistent pain and inflammation. The Provant therapy system provides a non-narcotic option for these patients through its anti-nociceptive analgesia and anti-inflammatory effects. Provant increases endogenous opioid expression to provide analgesia. It mediates cytokine and edema-related genes to help tissue move through and out of inflammation. And it mediates inflammation-related genes to allow for the resolution of inflammation. Provant therapy may be an option for you and your patients. You can learn more at regenesisbio.com.